Today, we are looking at the effect of pH on the activity of amylase. So, we'll start by saying the potential of hydrogen, which is the pH, is the best measurement for determining the concentration of hydrogen ions in a solution. The pH also determines whether a medium is acidic, basic, or neutral. So what we have to note is that mediums with pH below 7 are called acids, whereas mediums with a pH above 7 are called bases or alkaline, and mediums with pH 7 are called neutral. So from our chart over here, we can see that a solution with pH of 7 is termed as a neutral, and with a solution with pH lower than 7, that is from 0 to 6, is termed as an acidic solution. And mediums with pH greater than 7, that is from 8 to 14, are known as an alkaline solutions or medium. So first of all, what we also have to notice is that enzymes are basically proteins composed of amino acids that contains acidic carboxylic groups and basic amino groups. And the presence of these groups, that is the acidic carboxylic groups and the basic amino groups, make enzyme to be affected by changes in pH values. So as we can see, this is an example of a protein over here with the carboxylic groups and the amine groups over here, and this is the peptide bond. Also, each enzyme has a pH value that it works at with maximum efficiency, called the optimum pH. So if the pH is lower or higher than the optimal or the optimum pH, the enzyme activity decreases until it stops working. So let's look at some example. Example, pepsin works at a low pH, that is, it is highly acidic, whilst trypsin works at a high pH, that is, it's basic. So let's take a look at this graph over here. So this is a graph showing us the effect of pH on enzyme activity, and we are going to use three different enzymes: so the pepsin, the salivary amylase, and the alkaline phosphatase. So from the graph over here, we can see that the enzyme pepsin has an optimum pH around 2. The salivary amylase also has an optimum pH around 6 to 6.5, whilst the alkaline phosphatase also has an optimum pH around 9. So what it means is that salivary amylase will not work at a pH 1, and pepsin will also not work at pH 9. Optimum pH for salivary amylase. The optimum pH for the enzymatic activity of salivary amylase ranges from 6 to 7. Above and below this range, the reaction rate reduces as enzymes get denatured. The enzyme salivary amylase is most active at pH 6.8. So let us now look what an enzyme activity is. So we define enzyme activity as the amount or moles of substrate converted to products per unit time. And enzyme activity helps to analyze the number of substrates converted to products and also to analyze the enzyme efficiency. Let's look at the materials and the reagents that we are going to use to perform this experiment. The first has to do with the salivary amylase, which is our enzyme, our buffer 0.1 molar phosphate citric acid, and our 0.1 molar phosphate buffers. We have our iodine solution, we have our 2% starch solution, And we also have our water bath set to 37 degrees Celsius. So before we start, one thing that we have to note is that we are to obtain at least 6 ml of saliva in a test tube in the morning by chewing an inert material to help produce enough saliva. And also, you are provided with the following buffers, each containing 0.5 molar sodium chloride. And the importance of the 0.5 molar sodium chloride is to act as an activator. So from the table, we have our buffers. We have our buffers here. So which are the 0.1 molar phosphate citric acid buffer of different pH and our 0.1 molar phosphate buffer of different pH. So as you can see over here, for the 0.1 molar phosphate citric acid buffer, we have different pH values of pH 5.0, 5.5, 6.0, 6.5. And for the 0.1 molar phosphate buffer, we have different pH values of 7.0, 7.5, 8.0, and 8.5. So to start with this protocol, we first have to prepare a substrate buffer for each buffer from the table above by adding 5 ml of our 2% starch solution to 5 ml of each buffer in their respective labeled test tubes. So for test tube 1, we have 5 ml of the 2% starch solution and 5 ml of our 0.1 molar phosphate citric acid buffer of pH 5.0. For test tube 2, we have 5 ml of our 2% starch solution and 5 ml of our 0.1 molar phosphate citric acid buffer of pH 5.5. For test tube 3, we have 5 ml of the 2% starch solution with this buffer of pH this. For test tube 4, we have 5 ml of our 2% starch solution with 0.1 molar phosphate citric acid buffer of pH 6.0. For test tube 5, we have 5 ml of our 2% starch solution plus 5 ml of our 0.1 molar phosphate buffer solution of phosphate buffer of pH 7.0. For test tube 6, we have 5 ml of 2% starch solution plus 5 ml of our 0.1 molar phosphate buffer of pH 7.5. For test tube 7, we have 5 ml of the 2% starch solution and 5 ml of our 0.1 molar phosphate buffer of pH 8.0. And for test tube 8, we have 5 ml of the 2% starch solution plus 5 ml of our 0.1 molar phosphate buffer of pH 8.5. And what we have to note is that this diagram over here represents each of the test tubes containing our substrate, which is the 2% solution, and each buffer at the different pH values. So the next thing is to equilibrate each substrate buffer and saliva, which is the enzyme, in a separate test tubes in a water bath at 37 degrees Celsius for 5 minutes. So this is our substrate buffer, and this is our enzyme, and this is our water bath set at 37 degrees Celsius for the equilibration. And as said earlier, the substrate buffer over here represents each of the test tubes. The next step is to perform an enzyme substrate reaction mixture by adding 0.5 ml of the equilibrated enzyme to our substrate buffer in a water bath set at 37 degrees Celsius. So the next step is to perform an iodine starch test by spotting on white towel or spotting towel until reaction is complete. So We do this by just picking a drop of our substrate enzyme mixture in the water bath and adding it to a drop of iodine solution on a spotting towel. And what we also have to note is that we also have to prepare a positive control and a negative control test for this, for the test reaction. So the positive control is basically a spot of the iodine and a spot of the 2% starch, and the negative control is a spot of the iodine solution. And what you also have to note is that we have to repeat or perform this iodine starch test every minute until there is a complete reaction. So as you can see over here, at 0 minutes, this is a reaction. 2 minutes, 3 minutes, 4 minutes, 5 minutes, 6 minutes, 7 minutes, and 8 minutes. So these are all reactions for each of the time over here. So what you have to note is that reaction becomes complete when the blue-black color formed at the initial stage, that's starting from the first spot, turns completely to yellow or start to produce a yellow color in sequence upon the addition of substrate to iodine with regards to the time interval. So let's just study this diagram over here. So we can see that at time 0, we have this color. At time 2, 4, 6, 8, 10, and 12. We have different colors over here. But at time 14, we have a yellow color over here. So what you can say is that at time 14, the reaction turns completely to yellow and start to produce a yellow color in sequence upon the addition of the substrate enzyme reaction to the iodine. So if you can see, from time 14 to 20, we have what? A sequential yellow color being produced over here. Hence, we can say that at time 14, the reaction is complete for this test. And what we also have to note is that if the reaction is very slow, conditions can be altered. Example, the amount of enzyme can be increased. The same and the same must be done in all tubes. And also, if the reaction is too fast for accurate measurement, dilute the saliva or the enzymes. So this is our assumed table of results. So as you can see over here, the table is showing us the various buffers with their respective pH and the time that it took for a complete reaction to occur. So you can see that at pH of 5, the time for complete reaction is 36, 5.5 is 29, 6.0 is 20, 6.5 is 10, 7.0 is 12, 7.5 is 17, 8.0 is 25, and at pH of 8.5 is 33 minutes. So let us now try to calculate the rate of reaction in milligrams per mole per minute. Example, calculate the rate of reaction at each pH, and the unit that we are going to use, milligrams of starch broken down per ml of saliva, and plot a graph of rate of reaction against pH. So first of all, what we have to notice is that we use 2% starch as our substrate in this reaction, and the 2% starch is equal to 2 grams of the starch in 100 mils. But we also have to notice that we use 5 mils of the 2% starch as a substrate in the reaction. So we will now determine the grams or the mass in milligrams of starch in the 5 mils of the 2% starch used as substrate. So what we are going to do is that we are going to equate the 2%, which is 2 grams equals 100. So if 2 grams is equal to 100, which is for the 2%. But then how many grams of starch will be in the 5 mils of the substrate that was used? So making as a subject over here, we'll get S in grams to be 10 divided by 100 over. So we'll get 0.1 grams over here. But we are to express our result in milligrams per mil. So what the next thing to do is that we will convert the 0.1 grams into milligram. And if you are to convert this into milligram, we will get 100 milligrams. So rate of reaction at each pH is given as the amount of starch in milligram divided by the volume of enzyme used to break the starch times the time taken for complete reaction. And what we have to note is that the mils of starch used over here, or let me say the amount of starch used in this test is the same for each of the buffers, and the volume of enzyme used to break the starch is also the same for each of the pH values. So at pH of 5, our rate of reaction will be the 100 milligrams, all divided by 0.5 mils, which is the volume of the enzyme used and the time taken for complete reaction. So if you are to solve this, we will get 5.6 milligrams per mil per minute. So at pH 5.5, the rate of reaction will be 100 milligram divided by 0.5 mil times 29, and our final result will be 6.9 milligrams per mil per minute. At pH of 6, we have our rate of reaction to be given as 10 milligrams per mil per unit. At pH of 6.5, we have our rate of reaction given as 20 milligrams per mil per unit. At pH of 7, we have our rate of reaction given as 16.7 milligrams per mil per unit. At pH of 7.5, we have our rate of reaction given as 11.8 milligrams per mil per minute. At pH of 8, we have our rate of reaction given as 8.0 milligrams per mil per minute. And at pH of 8.5, we have our rate of reaction given as 6.1 milligrams per mil per minute. So this is just a table to show the pH values that we have, the pH for each buffer, and the time it took for a complete reaction, and the calculated rate of reaction in milligrams per mil per minute. Now we are to plot a graph. We are to plot a graph so now we are to plot the rate of reaction against our pH values over here. So to plot the rate of reaction against the pH value over here, this is the nature of graph that we are going to get. And from the graph, we can see that the optimum pH for the salivary amylase is 6.7. Thank you.